So, through this, from the beginning, we've been talking, we started about ecology, we're talking about looking at communities and stuff. And you heard a little bit from Farah how it's also possible to investigate a little bit more about the vectors and their dynamics through genomics. You heard from Chitra about pathogen genomics and Rajesh, like how to sequence genomes of pathogens, how to investigate their pathogenicity and so on. So today's talk is by Dr. Balaji Chattopadhyay. He's a faculty at Ashoka University. And he has actually spent a lot of time working on bats. I would probably say he's one of the few people in India who really understands bats and their distributions. From his PhD days, he's been working on bats and also on genomics. So he's going to talk to us about host genomics. So not pathogen genomics, not vector genomics, but host genomics. So I hope he's still here. So thank you so much for coming, Balaji. He had to take a session earlier, but he was unwell. But he kindly agreed to come today. So thanks so much, Balaji, and look forward to your talk.
Okay, so good morning or good afternoon, everyone. Basically, I was not supposed to give this talk. I was supposed to do something else, right? So this is a kind of impromptu presentation, hence it's a bit scattered. I just put some slides together, and what I want to narrate today, basically talk to you guys about, or rather interact about, is, we talk about pathogens, we talk about infections, we are talking about zoonotic pathogens, vector-borne infection, infectious diseases, many things that actually is spilled over from wildlife. But how much do we know about the wildlife? How much do we know about the ecology, about the biology, about the evolutionary trajectory? What might be the conditions that might have accelerated or expedited, particularly in evolutionary terms, such spillovers also harboring pathogen. Also, how do they tackle infections? So this is a vast field currently. It's exponentially growing for past decade or so, and those who work on bats, I guess they are kind of in the forefront of this, because as you see more and more pandemics, epidemics and pandemics, and we are going back to wildlife, and many times we are actually going back to bats as we are trying to look for a source, right, to blame. Now, bats comprise almost about a fourth of, I guess, all mammals, close to around 1,500 species. India is actually quite diverse in regards to bat diversity. There is more than, this is a very rough estimate, so hence I put approximate. I still don't know the exact count. I guess in a year or two we'll have an idea. 10 years back, it was 120 species. Now we believe it's more than 130 species of bats. And we are finding more. I will get into some of those things also as part of my talk, that how frequently, we go, we catch, it's new, right? And that has great implications, because the pool of microbiome many times can be species specific, right? And similarly, the mode of transmission, then we need to know, once we know what the microbiome is. Anyways, so that's how, in a way, the diversity of the host in regards to number of species or number of unknown species that we are trying to find might contribute. So they have also high cryptic diversity, because for many bats, using morphology, you can't really ID them. So when you catch them, when you hold them, right, when you measure them, many times they all look similar, but when you go and do a barcoding with cytochrome B or CO1 gene, you find that, oh, they are vastly different, right? And the other important is they are very sensitive to changes in climatic patterns, and hence they are bioindicators. Of course, they are keystone species to ecosystems. I don't have to reiterate that. In regards to bioindicators, I like to say something, because as their sensitivity adds actually also to their susceptibility to infections, as we come to know. you would read reports year after year during summer months in Australia of flying foxes in Australia dropping dead in thousands. Fine. Similarly, in Sonipat, in my campus, this year, it was extremely hot. And we were finding bats dropping dead from nowhere. There were no bat colonies in our campus before. We suddenly had insect bats coming in, taking refuge, dropping dead in the middle of the day. And we have seen this like multiple deaths. So it's very real, and a very proximal thing like climate change, warming, extreme weather conditions can have great effect in the physiological conditions and can actually facilitate pathogen movements. And so just to reiterate, this is the Oriental realm, or some of us might also call it Indo-Malayan realm. And of course, the warmer colors are where you have the highest diversity. India has fairly high diversity, particularly if you look at Western Ghats and if you look at Northeast, even in Western Himalayas. Western Himalayas, more and more we are sampling, this will increase. Eastern Ghats, not well sampled, even Peninsular India. Overall, we believe India actually has a lot of diversity that we still don't know of with bats. And this kind of also correlates, I don't have the map here, this also kind of correlates with predicted hotspots for zoonotic outbreaks. So the predicted hotspot for zoonotic outbreak is also southern China, Southeast Asia, and South Asia as well. So now we know that bats, already this is not news to any of you, and many of you actually will be more knowledgeable than me in this regard. They harbor high diversity of viruses, and particularly for RNA viruses, very high diversity. We believe much of this is due to focused assessment, and we are actually looking for RNA viruses, so we are finding more and more of these. And we also know that bats are largely asymptomatic, right? So some of the things that a lot of people some of the questions people are trying to address is, why, even though they have high diversity of viruses, what are the processes, what are the immune system, how it works, that actually ensures that they remain largely asymptomatic. So they clear infections really fast. It's not like they are not infected, but they clear infection very fast and they suppress inflammation, right? And this is predicted paleotropics, particularly Southeast Asia, South China. This zone is predicted to be a hotbed for such outbreaks. And as we know that there are multiple episodes of recurrent outbreaks for past 2 decades and so. So therein comes integrated field, which I did not know much about 10 years back, but now I align myself with it, supposedly. It's called biodiversity genomics. So where basically anything that entails genomics or multiomics data and you look at biodiversity falls within this paradigm. This field also aligns very closely with One Health, wherein you are now able to look at animals as hosts, as reservoirs, and how that, the entire dynamics of how it finally comes and affects human health, right? So some of the components that we do are genomic surveillance, where we are looking at discovering pathogens and monitoring them. We look at host diversity. That is what mostly that I will be talking about. We look at host diversity, not only about diversity of number of species or their abundance, but also genetic diversity within species. We look at phylogenetic. So evolutionary history of pathogens, how you already learned some epidemiological models and all, how such models are applied actually with genetic data, genomic epidemiology basically. We also look at host pathogen coevolution. So we learn actually, because hosts have been exposed to pathogens over their history, right, of existence. So for tens of millions of years, for example, bats have been exposed to various pathogens. And they had the opportunity actually to evolve. The pathogens also had the opportunity to evolve strategies to infect. Bats also had the opportunity to evolve strategies to avoid some serious infections. So some of the research themes that falls under this, one is, go and, so basically some of these are the starting points. Go and discover what is there, like, is there a new species? Is the species isolated, then get into their pool of microbiome, find out among the microbiome pool what pathogens you're looking at. Another aspect will be to understand how these population and species are responding to climate change, habitat alterations, both ancient as well as recent. A third component is actually to look into host genomes, to actually find out all these, to look into all the strategies, particularly how the immune system might have evolved and all over the course of tens of millions of years, right? To avoid or to fight pathogenic infection. So part of some of the work that we do is actually discovering biodiversity. This started very long back, and this started actually in Uma's lab back in 2008 when Kritika actually joined Uma. And I had a new, like this is called phonic type. I had an insect bat from Yerkad which was emitting a different call frequency. It looked the same as a bats of the same species from Mahabaleshwar, but it was emitting a different frequency. And we wanted to actually do sequencing, and Kritika had just joined Uma for PhD, and she said, let's do some SIDB sequencing. We found that these are distinct species. We are still continuing with this. Two species. That point of time, it was mitochondrial. Later, we verified with few nuclear genes. They are still different clades. They are quite distinct. Now we have actually whole genome shotgun sequencing data from several animals, right? So, and we plan to do actually the high quality whole genome also for one of these species. Similarly, we went to Western Ghats a few years later when I joined Uma as a postdoc. I found another new species of insect bat in the Western Ghats. Once I joined here for PhD, once I joined here for faculty, I got a project to look into the diversity and do genomes from bats from Northeast. We went and we actually caught a bat. It's a greater horseshoe bat, has a huge distribution from UK to Japan. And there are multiple lineages. And as of now, we know that several of such lineages are actually species level lineages. We found that a sample that we got from Manipur was pretty much distinct in mitochondrial genome and as well as its nuclear genome for the same subspecies level lineages that's found adjacent in South China. So it's a new discovery. It's a new species level lineage that we might have bumped upon. Same thing, around the same location, we found some bamboo bats, and we said, okay, we'll do the genome of bamboo bats. We found that this bamboo bat is very distinct from any other bamboo bats of its lineage. It's a new species. Our collaborators around the same time have also found new bamboo bat species from Kerala, from Western Ghats, which is again very distinct. Going to a more encompassing geography, during my postdoc with Uma and further in National University of Singapore and still continuing work, we are looking into this fruit bat genus called Cyanopterus, which is like present everywhere. And using next-gen sequencing and genome-wide dataset, we were able to find out that across this region of fruit bats, there are various, not just distinct lineages of distinct mitochondrial lineages, but also corresponding distinct nuclear lineages as well. So basically, there's a lot of cryptic diversity, particularly specific to some islands. For example, this lineage is specific to Philippines. It's present only in Philippines. We have a lineage which is present only in Sulawesi. We found a lineage which is present only in the island chain of Palawan. That's between Borneo and Philippines. We found another distinct species level lineage in the Sundaland island populations. And we also, as we are looking into it, we also observed that there was some mitonuclear discordance. So when you do a nuclear gene tree using our genomic data set, and you have these individuals here for different lineages, for the same individual, for example, for this individual, which is a minutus, the mitochondrial DNA is actually from princeps. It's from a different species. So there has been integration events that people have missed out. We further went and actually quantified what kind of nuclear genomic integration these various island lineages are facing. And what we observed is that this lineage of minutus almost has 40 to 50% contribution from another lineage, Synapterus pinks. Similarly, this new species lineage from Palawan that we observed seems to be a lineage which has almost equal contribution from the Philippine species and from another species from Sundaland. Now the question is, why should I be interested in understanding these dynamics? When I want to only look into pathogens. Of course, I don't want to only look into pathogens. What happens is that, through gene flow and through integrations, genes, segments of genomes are being exchanged, right? For example, for our data set, Kritika can correct me, we found genes responsible for embryogenesis that have been exchanged, right? But it is also possible that genes which are related to defense mechanisms can also get exchanged, right? I'm not saying it is happening, I'm saying can, right? So therein lies propensity of movement of genomic components between species through integration. There is also a propensity of movement of pathogen, not through integration rather, but I'll get into through integration also there is a possibility through a different way, but generally through cohabitation and wherever there is actually opportunity for admixture, where two species are actually meeting, even though they have their unique gene pools, under certain ecological conditions or under certain conditions when they meet, when they interact, there is a possibility of movement of pathogen between them as well. So the more, a more indirect thing that we do is actually to understand response of these bats to climate change, and that's ancient. That happened, hundreds of thousands of years back, tens of thousands of years back, not very recent. And that you can do actually from a single genome. You look at the heterozygosity across the single genome, and you will be able to construct or reconstruct the population size of the species going back to millions of years. And if you are able to reconstruct paleohabitat or paleo niche, you will be able to figure out if there is a connection for that particular species in change in population size and change in its suitable available habitat. So while we are up to this is an old study, we are continuing with a much larger panel now. So while we are up to this, what we found is that for about a dozen species, that point of time for which there were genomes available, frugivores, whatever were there in the panel, they were pretty susceptible to global warming. So whenever there was a warming period, this last interglacial was a warming period, right? Whenever there was a warming period, the population size was low. And as we are getting into a colder period, the green dot, what is the last glacial maxima, population size increased. And this kind of was a common theme for a few species that were in our panel. For insectivores, we had variable information. For large insectivores that we had in our panel, we found out that the usual thing is that they have very low population size. However, one common theme that was there, that as they were entering Holocene, so around 12,000 years back or so, as they were entering Holocene, their population size or genetic diversity was actually pretty low historically. So that made us infer that mostly bats actually have been sensitive to generic climate change. So maybe even though a bat is, currently we say that it's a specialist or it's a generalist, it's endangered or not, overall, most bats might be actually sensitive to vagaries of climate change. And the population size estimates that we got for bats as they were entering Holocene actually were comparable to endangered carnivores. Okay, so currently we have expanded the work to about 3 dozen of bat genomes, and we are seeing the similar pattern. So we wanted to know, it was a very smaller panel, let's increase the panel. We are seeing a similar pattern, and we are again and again seeing that as
Sorry. Yeah. Balaji, maybe you can just briefly explain what is PSMC or how, because they've not come across, just like how you can use those data to look at population size. Okay. Not the full form of PSMC, but.
Okay, fine. So basically what happens is your genome has contribution, the nuclear genome has contribution from father and mother, right? So you are getting 2 components, 2 alleles, one from father, one from mother. And that gives you an opportunity to have some variability throughout your genome, but the variability is not actually similar across the genome. So what this program does is actually looks into this variability into different segments of the genome. Now, the fact is that if father and mother are closely related, right? If they are coming from an ancestor which is not far back in time, in that case, the variability between father and mother, the components that you are getting, these two alleles, will not be very high. Fine? So you are finding a common ancestor very recent in times. And if for many fragments of the genome, you are again and again finding common ancestor in recent time, which means that there were very few ancestors at that point of time in the population, right? Which actually gave rise to few offsprings, and as such, whatever the gene pool that you see now is coming from few ancestors from that point of time. So every segment in the genome gives you such alternative or such differential understanding, right? So that component is actually taken up in this graph. So for this particular bin, this is 10,000 years, this 100,000 years. So for this particular bin, what it says is that the population size was pretty low, right? Because there were many such locations in the genome wherein common ancestor was found here compared to maybe this part or this part, right? That's the information that flows through the diagram. Okay, so now this is like how they responded to ancient climate change. This kind of gives us something to assume that if they are sensitive to ancient climate change events, maybe they are sensitive to what is ongoing. Habitat destruction, global warming, maybe they are all sensitive to what is ongoing. That we were able to actually test with one of these fruit bats. So the Anthropocene, particularly for the past few centuries of industrialization, is basically characterized by vast amount of extinction of wildlife. And what is common knowledge now is that we are losing diversity faster than we are actually discovering them. And what we were trying to ask is that, if biodiversity is actually being lost, species are under stress over centuries and centuries, over generations after generation, if their sizes are going down, do we see the signature in their genomes? So for this, we actually chose Singapore as an island system in one of our earlier studies, where around 1819, it was mostly inhabited, it's all green and all, but then in 1990, there's no greenery. So 90% of the forest cover is lost in a century, and up to almost 87% of its biodiversity was lost. We chose a fruit bat, it's called Sunda fruit bat, Synapterus brachyotis, which is synanthrope. It likes to live close to human habitations. It doesn't live in forests. So whenever there is a primary forest patch, you don't see the bat, you see a different species. This species only likes to live close to human habitation and degraded landscapes. So again, we collected genome-wide data, and here what we did is we took degraded samples from a population that was collected in 1931 and was stored in Singapore Museum. So we took samples from them, and so it's a temporal sampling. We took samples from there, that point of time, and we took samples from 2011, 2012, which were really fresh. And we knew that, around 1940s, 1950s, urbanization really started pace. So post Second World War, urbanization really started taking shape in Singapore, and a lot of destruction happened. When we looked into overall diversity, genetic diversity or heterozygosity within the genome of these bats, we found that within the fresh samples, the diversity is actually pretty low compared to the older samples. Now, then we actually tried to look into, if the population experienced some drastic decline in past, 100 years or so. And therein what we found was that the population actually declined almost 30-fold. And it happened around the 1940s, even though the population actually was declining much earlier, but a major bottleneck was experienced around 1940s, 1950s, and the population has not recovered since. Okay, so these are some of the things that we were accumulating information that bats, one, you have distinct species, there's a lot of opportunity for gene flow, and also they seem to be sensitive to climate change, and some of them we see that certain signature of declines associated with urbanization. Now, in addition to that, we said, okay, fine, let us look into bat genomes and let us see how some of these genomes, what kind of diversity they harbor. Particularly, we are looking into immunological diversity, right? So, for the same Sunda fruit bat, we actually did a whole genome quite a few years back, and what we found was that similar to other bats, they actually experienced a lot of fluctuations in various immune-related genes, particularly some of the genes that are responsible for upregulating inflammatory pathways. They were absent or they were highly suppressed, right? Then there were some of the genes which are supposed to actually fight viral infections. They were more in numbers. And for a few of such families, I don't recall the names exactly, for few of these gene families, we saw expansion. For few, we saw actually contraction, mostly in line with what we were finding with other bats as well. In general, what we know that bats actually use diversifying strategies. No two bats use exactly the same strategy, at least that's what as of now we know. They use various strategies, but a common theme remains that, one is, an action against virus, and the other action is to suppress inflammation. So because you suppress inflammation, your the fever is down, so you're vastly asymptomatic, and your action against viruses, either killing them or removing them from the system sooner, actually gives you more opportunity not to get severely symptomatically infected. Anyways, so we are continuing the same theme, back in India, and here we got this new species, high quality genome of the new species now, wherein the species has about 28 chromosomes. We have been able to reconstruct pseudochromosomes for 23 of these. We are still correcting errors in the genome. And then for the new bamboo bat species that we have found, for that lineage also currently, we are looking into the genome. And we'll go back and we'll perform the same analysis, particularly for the horseshoe bats, because horseshoe bats, they harbor tons of viruses. So the next genome that we are going to do currently that we are doing is that of the other cryptic species that we discovered earlier, Rhinolophus rooksii. They are quite unique because they harbor a lot of viruses and their immune system also is quite diversified. Okay, so now that was more about what we are looking at hosts, and we can look at host genomes. We can say that they have these diverse immune strategies and things like that. Now we'll get into something more directly relevant to pathogens. These are called endogenous retroviral elements. So retroviruses gets integrated into host genomes. Fine. You have HIV. I think there's a lymphatic virus also in humans. You have a couple of zoonotic viruses, right? And we know now that retroviruses are present in almost all vertebrates. They kind of originated, I guess, in ocean some 400 or 450 million years ago or something, and they have been passed upon, and vertebrates have been continuously experiencing retroviruses, fine, and they have been evolving along with retroviruses. Also, retroviruses get inserted into the genome. They become what we call genomic fossil. So they may actually give us truncated products or no product at all. There will be stop codon or something, right? As time goes on, generation passes, as they pass through species and events. Not all retroviruses actually gets incorporated and what we call endogenized. Generally, retroviruses come and attaches into somatic cells. Very rarely, these actually pass on to germline cells and pass on to the next progeny. When that happens, we call that endogenization. And more such endogenization events can happen, and after more and more generations, they can actually take up very specific function for which they were not meant to be. We call that X-adaptation. So one is something called synctin, if I remember correctly. it's a barrier in your placenta actually to save the fetus in humans actually from pathogens and other components from the mother. That actually is coded by the envelope protein of one of the retroviruses through endogenization. You have genes, again envelope genes of retroviruses in animals and I guess also in humans, whose truncated products confers antiviral functions, antiretroviral functions to the genome. So the host actually has developed antiretroviral functions associated with endogenization of this envelope protein from a specific retroviruses. So under such context, we realize that in Indian landscape, it's very difficult to actually go look at, it's a lot of funding, permits, everything is required. I can't do host genome, look at host population level variability, their functional diversity, and get into their pathogen pool all together at one point of time. It's very difficult. So we said that, okay, a convenient way to enter the field is actually to look into these endogenous retroviral elements, ERVs, and look at their evolution, their pattern of diversity in the genome, their evolution in bats. And for this, we did not really have to go and do a lot of fieldwork or anything. At that point of time, around 43 bat genomes were publicly available. We downloaded the genomes, we found out what all retroviruses were there, and we did some simple analysis. So one thing we found was a positive correlation between the genome size and the number of endogenous retroviral elements that they harbor. A few other things that we observed is high diversity of two genera of retroviruses, gamma and beta, and then a particular genus of retrovirus alpha, which previously was commonly found in birds, we found in lots of bat species. We also observed more incidents of rare ERVs like lenti and spuma in bats. And then we were asking, okay, the way we look into or mine these retroviruses is basically there's a panel that we know, okay, so, this is a retroviral sequence for this, for example, envelope protein. Now, we tell this panel, okay, fine, find in the genome if you have a similar match or not. Once there's a similar match, we take the similar match and we go to a public virus database and find out if this similar match actually matches with a retrovirus in the public virus database. So we are confirmed now this location in the genome has an envelope gene which is of retroviral origin, right? But then how do we know they are functional or not? They are genomic fossils or they're intact? They can code for all the envelope proteins or not, right? We don't know. For that, what we do is we find out basically the start point is that, is there a stop codon in this sequence that I actually just mined? So when we did that, we found that nearly 30% of all the indigenous retroviral elements that we found did not have stop codon, they were intact. But 70% of them were like really fossils, they had stop codons. So even if they were products, the product will be truncated or there will be no product. So the other thing that we did is that as we are doing the study, a paper came out, not this one, okay, the reference is not there. A paper came out where, which actually established a database based on machine learning. And they identified all viruses as having high, medium, or low zoonotic potentials, including retroviruses. So we actually matched our ERVs to that particular database, and we found that almost all species in our panel harbored some or other retroviral elements that is predicted to be of high zoonotic potential. And particularly, this was enriched in 6 species of bats, 5 of which are from paleotropics. Yes, Uma.
So basically what you're doing here is you're using these retroviral elements as proxies for kind of evolvability. Yes. Or are you, is that correct?
Kind of evolvability. So the database is actually, it's a predictive database, right? Because we don't really have a lot of retroviruses that we know are zoonotic or high zoonotic or less zoonotic. So the database is a database of all viruses. Based on that, it predicts. And based on how close, it's a phylogenetic affinity kind of thing, how close our elements are to these elements, known elements in the database, they are actually assigning it as, this is a predictive thing, and this is just a start. A good research actually, or a real good research actually will be to test these in cell culture systems, develop actually experimental assays.
Yeah. So, for example, you guys all watched that recording, right, by Rory. So he talks a lot about hazard, and one of the slides or two of the slides are actually saying how do we predict potential pathogenicity. So this is another kind of route for that. Just to flag that.
Yeah, so I just realized that I have finished with my presentation and pretty fast. So I'll just then conclude. Yeah. Okay. So now the thing is big question. As Kritika helps me compile all these things in general, and we were discussing last night. So how can with an audience like yours, how can we put forth an argument that all these things that we are talking is actually connected to pathogen genomics, right? So I already tried to put forth the argument in favor of that. I'll just repeat. So one is, when we identify past infections, not only we know how groups of animals have been evolving with such infections, we also find out how they develop new functions, right, as they are exposed to such infections. So through comparative genomics, again, we actually are able to get more and more in-depth information, particularly for your context, immune system evolution, how diverse it is, how it functions against various pathogens, etc., etc. So coevolution of host immune systems to tolerate infections. Then as we are looking into climate change aspect of the host and all, we can ask distal and more proximal questions. How association in loss of genetic diversity, it's a vast field, right? How is there association between loss of genetic diversity or loss of gene flow, etc., to susceptibility to pathogens, to its pathogen pool and pathogen movement, right? Not only maybe within a same species, but within different species of cohabiting bats. Then going ahead with bats and non-volant mammals, then going little bit ahead from there between peri-domestic animals when they are in a degraded habitat, in more stronger cohabitation, from there going to domestic animals, and maybe if we are very fortunate, bring into human surveillance. Fine. So it's a long road, and that's the start point can be somewhere here. So again, population genomics and biodiversity discovery also feeds into this. So we can understand connectivity between host population, resulted pathogen sharing, and discover new species and their associated microbiomes. So anyways, that was in short what I wanted to talk about. And acknowledgement, I have my granting body ACRB, my university granting from TSB, and civic granting, this was during my postdoc, funding from NUS, and some of the museums which from where I regularly get samples, field museums and this Singapore National Museum. And my mentors and collaborators and friends, you have Uma, Frank, Kritika, Susan, Ian, Gavin, Joanna, Sigit, Srihari, and Vishnudas. And the list is growing, thankfully.
So, great. So we have time for some questions also. Maybe you can think of how you see this relating to what we've heard so far, or any questions. The stuff about bat species, it's actually, I mean, it may seem like, why are we doing this work to find species and stuff? But it's crazy, right? That we don't actually even know very well the mammals in 2024. And there's a lot of cryptic diversity among bat species. And this is really important, for example, when we're looking at coevolution between pathogens and their hosts. If we don't even know what the hosts are, it's something which can be quite complicated. Any questions?
Why you have selected, what is that? What you said, hemorrhagic or inflammatory genes of host which are altered during the period of evolution. So on what basis you have selected those genes rather many other genes which are
so we
correlating with disease?
Yeah. So what we do is we try to find out You have genomes. Genomes are long stretches of DNA, right? And you'll have genes there. So we annotate, we characterize the genome into these various genes, right? That are present in various loci, right? And once we have characterized, and when we compare various genomes, from there we pull out which are the regions in the genome. So this is not a very specific study. For a specific study, I would say that, oh, I am interested. These are maybe, I don't know, critical natural killer cells. Example, no, just example. That, yeah, natural killer cells, for example, they fight viral infections, right? Yeah. So maybe I want to know about the diversity of natural killer cells in bats, right? For that, what I will do is, then I have to go and only sequence natural killer cells. That is what is selecting. Here we call these scans. So we are not selecting anything. We are asking what is there that is very unique in the genome of these animals, right? And whatever we find, I am presenting you a very small, tiny bit of information of what we found, because the talk relates to more of pathogens. There was a point showing that the bat that we analyzed a long back, it was enriched for a gene that helps in repairing telomeres. So protecting telomeres is supposed to is thought to be important for a longer lifespan. And bats, they live very long compared to the body size. They're this tiny, but they can live 40 years or 35 years or 30 years, right? So that was a discovery. We had some other stuff also that we found. I just selected the information to present to you.
Is there any correlation of those genes, altered genes, with that of acts as a vectors to co-evolution? Is there any supportive correlation with that of host and the pathogen to coexist by suppressing the inflammatory genes? So therefore,
no, for this I don't have an answer. For the ERVs, as I mentioned, for the ERVs, I know there is a correlation, because a specific retrovirus, once it gets endogenized, there are examples in rodents, I think in humans and few other animals as well. They confer resistance to the host, to the same retroviral infection again. That is there. For the first part, I don't know. Maybe people have found, but to the best.
There is work with Rusitis now that's just come out, where they've checked things with respect to their, so all of these interferons. So within fruit bats, you have a variety of interferons, and the way they use them is different. So Pteropus, it suppresses something. Rusitis has constitutive expression and things like that. So what they've done is they've looked at these MRSA virus, coronaviruses, and they try and see if it overexpresses in one cell line versus another cell line. So those things exist, and they all started off with comparative genomics by looking at which of these interferons are there, looking at their transcriptomes, what is overexpressed, what is not. And that's how now we are building up a database with organoid models as to why some bats are carriers of certain coronaviruses versus others. So, but it's very, very in the infancy. You have 1 or 2 labs who are able to do this, because these are all, again, BSL level 4 work. So very few labs do this, but they do this. It starts off from the genomics. So it's got a very long history of 10 years history of people doing experimental work and looking at genomic data to do this.
Yeah, I think I should mention this. So when I sample, I actually have a good PPE, right? When I transport, it follows World Health Organization guidelines. They're very stringent. And when I work in my lab, I actually work in a biosafety facility equivalent to working with, if you are doing SARS-CoV-2 work, so BSL2 plus, right? And I'm just doing host population genetics or host genomes. So anyways, yeah.
So Balaji, maybe you can give some idea on, for example, this cryptic diversity of bats that you guys have been discovering. It's kind of been, how should we say, what's the word? Serendipitous. Sorry, I don't mean it to sound bad, but you heard something like exploratory. You heard something, you found something sounding different. So, and a lot of work in biodiversity is like that. But I'm curious if you were to predict,
Yes,
like
so
if you
now-
were to actually do this study and say, okay, I want to find or better define bat diversity in India, in the Indian subcontinent, where would you look? How would you go? What areas, what taxa, like what families of bats? It would be great to hear from.
Exactly. So
Also, maybe, I don't know whether you have any slides on the bat families. That also would be nice to kind of show them.
no,
You don't have. That's okay.
that I don't have. There are like around 21 families of bats currently. I don't have the slides here, sorry. But to go back to Uma's question, yes, it started off as serendipitous, because it started off in a lab where one bat was echolocating strangely, and we are like, okay, so this is different, because for many bats, the echolocation frequency is tuned to the species. They are deaf to other frequencies. So we're like, okay, if this bat is deaf to the other population, then there's something going on. There might be distinct. And then we get into genetics, right? But then with one or two such discoveries, and when we are doing this, there was a group from Europe and ZSI that was working in Northeast. And they actually scooped us publishing before us. That was called Hippocidrus khasiana. Long back, I think 2009 or something. We published a year or two later. So we realized that, I gave you the context because we realized that diversity hotspots of bats, as of now, as we know, one is Western Ghats and one is in Northeast. And from that point of time, we are realizing that possibly these are the core areas where we can actually look. The question is why? Fact is, the kind of conditions that are prevalent in Western Ghats is pretty different from the kind of conditions that you find in Peninsular India, right? So if bats have to move, and you have the same similar species in Southeast Asia. So if bat species have to inhabit Southeast Asian monsoon and equatorial biome, and also be in India in more arid Peninsular India and more moist Southern India, you'll realize that there might be bats in the Ghats that might be more isolated, right? Because they might be used to the moist environment, and they may not be used to the more arid environment of Peninsular India. It's a hypothesis, need not be true, fine? Now you go to Northeast. Northeast also has areas of high rainfall and is connected, more importantly, to Southeast Asia directly, fine? So we were expecting to get more species of Southeast Asian affinity into Northeast, and we were expecting that we'll have more distinct species level lineages in Southern India. What we are seeing now as we have gone to Northeast is that regardless of its Southeast Asian connectivity pattern, we have distinct species level lineages in Northeast India compared to what we find in Indochina, right, or in Sundaland. So the level of diversity is more complex than than a very simple assumption.
So that is a really important point, because you think, like, we have been seeing all these meta-analyses, all these predictions, about how zoonotic risk is correlated with species richness or biodiversity, right? But all that is based on what we already know of what we think the biodiversity is. So how might some of this change? Like, this is really amazing, even for me, because I always think of Northeast as just like a comet out of Southeast Asia, of diversity. So nothing really unique. And so this is very, so how about, for example, within the Northeast itself, Himalayas versus...
Yes, so the other thing is, one is Eastern Himalayas and the Indo-Malay region. If there is any distinction, we still don't know. But what we assume currently is that between Western Himalayas and Eastern Himalayas, also there might be some distinction. So we believe that Western Himalaya, we have started sampling Western Himalayas, and we know that Rohit put forth some information as well previously. So we believe Western Himalaya also is going to be unique. But that doesn't mean that, Peninsular India is not unique. Peninsular India, we believe, might also be very unique. What I'm trying to tell is that over the course of millions of years, bats might have inhabited. These are all very different environments. They might have inhabited these different environments, and over generations, they might have gotten segregated enough that we can call them distinct species. Now you think, as they are segregating, their microbiome is also segregating. Some of it is generational. some of it is horizontal transfer. So they are accumulating unique microbiome. I am not saying pathogen also, right? Pathogen is part of the microbiome diversity, right? So therein lies the importance of understanding the host diversity. Start off with discovering what species are there, right? And then looking into patterns of connectivity, because through patterns of connectivity, there can be movement of pathogens. And even if there are, if there's an isolated population in an island, and even if there is an one-off migration event that brings in a new pathogen, within the scope of a small island, it can spread very fast within the island, right? So these are very intricately linked, but we don't really know much. What I am talking about, the things that I am talking, these are all hypotheses, and globally there are many labs where actually they are looking into all these hypotheses, testing them with much larger data set that we are thinking here. Yeah, there.
You have a mic over there? Yeah.
So within the insectivorous and fruit bats, the insectivorous bats are paraphyletic, right? Some of the larger, the rhinolophus and all, they clear with
That's sorted already.
Is there any phylogenetic signal to the viral diversity between them? I
No,
mean, they're closer to fruit bats, or
I,
that's
to
very
the best
different?
of my knowledge, no. What I know mostly is about some viruses are quite specific. I can't tell you what they are. I don't remember. But you have to understand the specificity of viruses in bats. It starts off with it can enter or not. If it enters, it can survive or not. If it survives, it can enter a cell or not, right? So all these things are important. Crossing the species barrier is pretty difficult, and hence for many cases, you won't have actually such movement. For some cases, we know there can be movement between species. That's what we try to check. When bats are cohabiting in a colony, will there be horizontal movement? Because they are emitting echolocation calls, right? So there will be droplets forming as they are talking to each other. And if they have active infection through saliva, they are urinating, they are defecating. Again, there can be droplets, so aerosol. Through saliva, it can actually, it's very rare, it can transmit, right? Now, even if it transmit, can it infect another species? So one basic assumption is that, considering all the cell receptor affinity and all that, is that if the species are phylogenetically close, then maybe there is more possibility for them to infect. If they are farther apart, maybe it's a dead end. But it's a very basic assumption. If you ask me, I really don't know the right answer. Yes, Ansil? No? Okay, fine.
So you're saying that, okay, I don't know whether I agree with you, but you're saying that still the most number of cryptic species will be discovered in the Northeast. That's kind of what you said?
No. What happened? So I am saying that when we went to Northeast, we were expecting Southeast Asian lineages or East Asian lineages. Fine. But then we end up finding something very different. Now it is possible what we are finding as distinct is actually present in Myanmar or Thailand or Vietnam, and they have not been discovered. That is also possible. That there are, and it's like because these are also monsoon lands. And then as you go down South Thailand, Malaysia, now that becomes equatorial, Sunda land. So the climatic conditions are also very different. So, and Myanmar is hardly explored. Similarly, part of northern Thailand and all, they are also not very much explored, even Cambodia, right? So as of now, we are finding something distinct, even though we don't expect. In the future, the assumption will stay or not, we don't know.
Okay, but I'm just wondering whether you think there might be cryptic species in more like arid parts of India as well.
Yes, I feel so.
Yeah.
I feel so. That is the thing. Nobody has done anything. We have money. No, sorry, we don't have money. We have we kind of have some permits here and there. I was trying to tell that we have permits. We don't have money. It's massive effort because particularly now, 10 years back, I could just go, I could capture, I could do a lot of stuff. Now it's massive. It's the amount of regulatory clearances I need and the amount of precautions I need to follow for all good reasons. It's fine. It's a massive effort actually to conduct field expeditions as extensive as to find. This is like, I am fortunate that I go and I find. I tell people, even though I go to a place where I know I might find something new, I have prior assumption before going, but I am still fortunate that I go, I find new stuff. But that may not happen always, right? So we need massive effort, and that will come with massive funding. I don't see the funding landscape as of now conducive for it.
Yeah, just to also bring up a point, like you guys will watch Rory, or you've watched Rory's video. So he works with this bat ecologist called Kate Jones. And, for a long time, I actually, until recently, people who worked on bats never really wore PPE, or, they never thought about it. And I heard an interview of, I don't know her, but I heard an interview of hers where she says she actually has this kind of chronic respiratory conditions, having, gone to bat caves for many, many years without PPE. So it's probably she has been exposed to something, and it's really important for us to,
Yes,
respect those.
I am also a living example of that.
If
Uma knows about
Balaji,
it.
he blames his respiratory conditions on Delhi, but I think it's probably the bat caves.
Yes, so I am also a living example, and any bat worker who does a lot of field work, catches bats, goes to caves, they will have a lot of respiratory conditions, particularly allergies.
Yeah, because also caves especially are concentrated environments. You're going into the cave, there's a lot of guano, it's a small space, there's not much ventilation. It's different from catching a bat in hand in field. This is, if you're going into caves, that's definitely a lot more potentially risky.
Scaring people.
No.
Sorry. Not scaring people at all.
Either follow the precautions or don't do the work.
There are many tourist places like Ajanta, and we have some places in Hyderabad also. There are many bats are there, and tourists are
so which means
also
that
there, so nothing is happening.
Yeah, which means that these events are extremely rare. I think Cliff was talking day before yesterday about Nipah, right? That these are extremely rare. So when my biosafety committee asked me this question, I tell that only, like, you have bats everywhere. They have been everywhere, all around. And if it was such a panicky situation, we won't be around at all, right? We'll be all dead, isn't it? But that doesn't mean that you don't follow precautions. No PP, no field work.
And yes, since we're talking about it, just to mention also, for example, our biosafety committee requires that all the people who work on bats have to have vaccinations, like rabies and so on and so forth. Every year, they have to have a health checkup, and that report has to be submitted to the committee just in case, something happened.
So, in that way, I think NCBS was a trailblazer, because when, like 12 years back, 13 years back, when NCBS asked us to vaccinate ourselves, and we were very happy, by the way. And no other place in India would actually ask, bat researchers to vaccinate themselves.
And I want to add one more thing, like concentration is also important. Where we work in lab setting, there we handle high volume and high concentration virus. There that PPEs are useful, but in field condition, it is not required actually. I mean, the PPEs are required, but the kind of PPE we use in laboratory are not required there. Like, there are asking too much.
You have no idea what kind of
No, but now it has become a requirement, and it's fine. It's better than not doing the work.
So here is the thing, what you are saying is right. The ideal scenario is that we are looking at bats, and they are not behaving strangely. That's what is the belief, right? So which means it's okay to handle them. But we still have to follow all precautions, that not only we get something from bats, but more importantly, we don't give anything to bats. That's a more important thing out here, okay? So we should not give things, because they are more susceptible, right? If we don't wear a PPE.
So it's about any animals, like, I work with carnivores, right? Of course, not with fecal samples when I'm walking in the forest, but if I'm interacting with animals, if you're interacting with animals, always good to wear PPE.
It's a good common sense. Of course, you don't have to wear like full Tyvek, Sugram, a lot of stuff and all sometimes. It's a good common sense.
Okay, sorry, that kind of went into regulatory stuff, but any other questions for Balaji? No? Okay, thank you so much, Balaji.
Thanks a lot.